In this video, we'll explore the key differences between peptide mapping and peptide sequencing. These two techniques are often mentioned together, but they answer very different questions in protein analysis. Let's break them down clearly and practically. This confusion usually starts with a very common situation in protein analysis. Someone asks whether the protein sequence was confirmed, and the answer is peptide mapping. The problem is, peptide mapping and peptide sequencing are not the same thing. To understand why, we need to look at what each method actually tells us, and what it doesn't. In protein analysis, everything starts with the question you're trying to answer. Usually, it's one of two things. Is this protein the same as it should be? Or what exactly is this protein made of? Peptide mapping answers consistency. Peptide sequencing answers composition. Peptide mapping works very much like a fingerprint. The protein is digested using a specific enzyme, most often trypsin. The resulting peptides are separated by liquid chromatography, often coupled with mass spectrometry. The pattern of retention times and masses forms a peptide map that reflects consistency. Peptide mapping is widely used in industry, especially in regulated environments. It helps confirm protein identity, compare manufacturing batches, and detect modifications like oxidation or deamidation. Because it is robust and reproducible, it's ideal for routine analysis. However, it is not designed for exploratory or unknown proteins. Peptide sequencing focuses on determining the actual amino acid order. Instead of asking whether something matches a reference, it asks what the sequence truly is. This is done using tandem mass spectrometry, or MS/MS. Peptides are fragmented, and the fragment ions are used to deduce the sequence. Peptide sequencing becomes essential when working with new or unknown proteins. It is critical when mutations or unexpected variants are suspected. Sequencing is also required when complete sequence confirmation is needed. This is especially important in early drug discovery and proteomics research. When we compare the two directly, peptide mapping is faster, more robust, and ideal for routine analysis. It is a comparison-based approach. Peptide sequencing, on the other hand, is more complex and data-intensive. It is discovery-based and requires advanced data interpretation. A common misconception is that identifying all peptides means the protein has been fully sequenced. That's not always true. Some peptides may be missing, co-eluting, or modified in ways that aren't fully resolved. Sequencing requires clear fragmentation evidence. Mapping shows coverage, but sequencing confirms identity. Peptide mapping and peptide sequencing serve different purposes. Mapping provides confidence in consistency. Sequencing provides confidence in identity. In many workflows, both techniques are used together. Choosing the right method at the right stage is what leads to reliable protein characterization.